transformation of fungi with exogenous DNA. So we have studied in previous lectures that the host organisms like bacterium, it is sometimes naturally competent. Like we have the idea that around 40 genera of bacteria, they are natural competent. Competent means that they have the ability to take naked or foreign DNA into the cell. And sometimes this competence or ability to take foreign DNA, it can be induced under laboratory conditions. So for bacteria, it is not so much problematic to induce competence in the cells. But yeast and fungi, they are naturally incompatible for transformation. So naturally they are not competent, and we have to use some specialized techniques for their transformation. Now, what type of techniques we can use? तो इस पर्पस के लिए हमें जो यीस्ट सेल है उसकी सेल वॉल को टोटली या पार्शली रिमूव करना पड़ता है. अगर तो यीस्ट की सेल वॉल जो है इसको पार्शली रिमूव कर दिया जाए तो एक स्ट्रक्चर हमारे पास आता है जिसको हम स्फीरोप्लास्ट कहते हैं. Now spheroplast, it is wall-less cell. So sometimes people can compare the protoplast where the cell wall, it is totally removed. And in case of spheroplast, some sort of differentiation is there, that here the cell wall is partially removed. So at the end we are going to get a structure if we are going to remove cell wall partially by using certain enzymes. So spheroplast is not there. Now this spheroplast, it can be, you can say that mixed with foreign DNA in the presence of calcium chloride, that can result in the uptake of foreign or exogenous DNA. Once the transformation is there, then such spheroplast, they again are cultivated on a semi-solid medium, like media that contain around 3% solidifying agent like agar. But this later step, that is the recovery of the cell wall after transformation, some people consider it inconvenient step. So because of this reason, now spheroplast, they can be exposed to heat current, so that by the process of electroporation, foreign DNA can be inserted or introduced into the cell. So electroporation technique along with spheroplast, it has been used for the transformation of yeast cells. In addition to that, some other techniques that are common in bacteria like conjugation, it can also be used. So for this purpose, a sort of shuttle vector can be developed that are replicating within the E. coli, and then they can be used to develop a conjugation tube with the yeast cell, so that foreign DNA can be inserted by using a sort of recombinant molecule. So to create a recombinant molecule, a sort of R751 plasmid or a type of F plasmid, they can be used, so that a recombinant molecule, it can be transported from E. coli to Saccharomyces or yeast or other fungi like Saccharomyces. Now, some other microbes, they can also be exploited to transform yeast cells. And here one can quote the example of Agrobacterium tumefaciens. ये जो माइक्रोब है एग्रोबैक्टीरियम, इसमें एक स्पेशलाइज्ड प्लाज्मिड होता है जिसको हम टी प्लाज्मिड या ट्यूमर इंडक्शन प्लाज्मिड कहते हैं. इसका एक पोर्शन होता है टी डीएनए जो कि एग्रोबैक्टीरियम से प्लांट सेल्स जहाँ पे वुंड डेवलप होता है वहाँ पे ये डीएनए को इंजेक्ट करता है। तो इसी तरह से जो टीडीएनए है, इट कैन बी ट्रांसलोकेटेड फ्रॉम द एग्रोबैक्टीरियम टू द yeast cell. It can also be used to transform hyphae as well as conidia. So these are a few techniques that we can use for the transformation of yeast and other fungi.